Hi everyone, my name is Riya Patel and I'm a junior at Rutgers University studying cognitive science with a double minor in biological sciences and health and society. This past summer, I had the wonderful opportunity to continue working in the Manzini lab at the Child Health Institute of New Jersey, where I collaborated with the Tischfield lab on a project regarding craniosynostosis and behavior. This poster, titled Exploration of Cognitive and Motor Deficits in Mouse Models of Craniosynostosis, summarizes my findings from this summer. Craniosynostosis is a condition which is characterized by an abnormal head shape and dysmorphic facial features, which is as a result of premature fusion of cranial sutures. While it only occurs once in every 2,500 births, there's an extensive list of neurological and biological complications, which requires us to learn more about and just understand the true extent of cognitive deficits. Previous research on craniosynostosis presented data of motor, social, and cognitive deficits in animals with a whole body knockout of twist1. Our hope in the lab is to study a single gene mutation to understand how the development of the skull and underlying vascular and neural tissues are integrated. Our gene of interest, twist1, it's a commonly mutated gene in many cases. It plays a key role in maintaining the boundary between neural crest and the cephalic mesoderm at the coronal suture, so a mutation in this gene leads to increased intracranial pressure. And this, in turn, affects brain function, which gives us reason to explore the cognitive and behavioral deficits in mouse models. So to do this, we used mice with a conditional knockout of twist1 in the cranial sutures to learn exactly how solely disrupting skull development impacts motor, social, and cognitive functions. So to begin with some preliminary data, we ran two common behavior tests, the wire hang and the open field. The wire hang primarily measures motor function and learning, and so a mouse is placed on the cage top, which is then inverted, and the latency to fall time is recorded. Our results showed that the mice cohort with twist1 conditional knockouts spent significantly less time hanging on the wire compared to the control group, which signifies a motor deficit. In addition, we ran the open field experiment, in which we placed a mouse in a chamber and allowed it to roam freely to get some data about exploratory behavior. However, this test gave us no significant results, which implies no specific deficits in regards to exploration. So using the results from these tests, we kind of concluded that motor deficits were present and they also required further research. So we continued to run behavior, this time with the novel object recognition test and the rotor rod machine. The novel object recognition test measures recognition memory and other cognitive abilities. It's split into three phases, and this test basically places two identical objects in the chamber and allows the mouse to become familiar with both of them before replacing one object with a novel object. Some data that we recorded from this experiment includes time spent with each object and a discrimination index, but again, there were no significant results. On the other hand, the rotor rod machine is used to measure motor learning and coordination, and this would actually help us see if there was actually motor deficits, which we had previously seen from the wire hang. As a baseline measure, we ran the machine with wild-type mice, which gave us the expected results of an increase in coordination and motor learning over the course of 5 days. Future research will continue to assess cognitive and motor deficits in older mice to understand how these cranial malformations affect brain functions. I would like to note that none of the behavioral tests we ran revealed any sex differences, but we will continue to assess any potential differences in our future research and experiments. I would like to take a quick moment and thank everyone at the lab, the Manzini lab, and the Tischfield lab for their support this summer and for the future. I'd also like to thank everyone at the Douglas Project and Douglas Residential College for offering me the resources necessary to excel during the summer and my time at Rutgers in general. And lastly, a huge thank you to my family and friends who have never stopped cheering me on in everything I do. Thank you so much.